Hello friends, in this video we will discuss about G-protein coupled receptor. G-protein coupled receptor is a membrane bound receptor. This is plasma membrane. You can see this is plasma membrane, and in this plasma membrane G-protein coupled receptor or GPCR present, and GPCR span plasma membrane 7 times. So this is called as membrane spanning receptor or seven membrane spanning receptor, or also known as serpentine receptor. But this receptor coupled with G-protein. This is G-protein, and G-protein is a type of heterotrimeric protein, and this is consisting of three subunit: alpha subunit, beta, and the gamma. So three subunit are there. When hormone is not binds with this G-protein coupled receptor, then the receptor remains inactive, and in inactive state the alpha subunit of G-protein binds with GDP, right? And when hormone binds with this G-protein coupled receptor, then the receptors will be activated, and this causes activation of this G-protein. And in G-protein you can see GDP binds with this alpha subunit in its inactive state, but this GDP will be exchanged by GTP, and after binding of this GTP with alpha subunit, the beta alpha and beta gamma subunit will be separated. Then this alpha GTP activate many downstream signaling molecule, and this signaling molecule may be adenylyl cyclase, or this signaling molecule may be phospholipase C. In case of group A hormone, this G protein activate adenylyl cyclase, but in case of group B hormone, this active G protein activate phospholipase C, right? So the G protein coupled receptor will be activated by two types of hormone: group A hormone and group B hormone. The group A hormone, in this case, catalytic unit is adenylyl cyclase. That means G protein activate this adenylyl cyclase, and the second messenger will be generated by this adenylyl cyclase. Okay, so the second messenger in this case is cyclic AMP. Noradrenaline, adrenaline, ACTH, LH hormone, FSH, GnRH, and HCG, and thyroid stimulating hormone. These are example of group A hormone. But in case of group B hormone, G protein activate phospholipase C, and the second messenger is IP3, DAC, and calcium. The hormones which are included under this group B are vasopressin, angiotensin 2, and oxytocin. Now we study function of group A hormone. When this group A hormone binds with receptor, this will activate G protein. So the alpha subunit which binds with GDP in its inactive state will be exchanged by GTP, and alpha and beta gamma subunit will be separated. Now this alpha GTP activate downstream molecule which is adenylyl cyclase, and this is also present in plasma membrane. This active adenylyl cyclase generate cyclic AMP as second messenger from ATP, right? And now this cyclic AMP enters into the cytosol, and this will activate protein kinase A into its active form. Then group A hormone always acts via second messenger, that is cyclic AMP, and this cyclic AMP activate protein kinase A. Now we see details about this G protein or G protein coupled receptor. GPCR is largest family of hormone receptor, and it spans cell membrane 7 times. So this is called as seven membrane spanning receptor or serpentine receptor. Then G protein is a heterotrimeric protein. You can find three subunit present in G protein: alpha and beta and gamma subunit. So this is heterotrimeric protein, and this subunit are different, so they are heterotrimeric. In inactive state, alpha, beta, and gamma subunit are combined together. So this is inactive state, and alpha, beta, and gamma subunit are combined together, and alpha subunit binds with GDP. So this alpha subunit binds with GDP. When hormone receptor complex is formed, the G protein conformation is changed. Then this, when hormone bind with receptor, then this active receptor activate this G protein, and in this case GDP of alpha subunit will be exchanged by GTP. So in inactive state GDP binds with this alpha subunit, but in active state this GDP will be exchanged by GTP, and after then alpha and beta gamma subunit will be separated. So this is alpha subunit, separated alpha subunit which binds with GTP, and these are beta gamma subunit. Now this alpha GTP activate adenylyl cyclase, and this will generate cyclic AMP from ATP, and this split ATP, and this forms cyclic AMP as second messenger. Alpha GTP activate downstream plasma membrane enzyme which is adenylyl cyclase, and this AC cleave ATP and generate second messenger, that is cyclic AMP. Cyclic AMP goes into cytoplasm and activate protein kinase A, and this protein kinase A elicit cellular responses. We study function of group B hormone, and in these cases hormone binds with receptor, and this will activate another type of G protein which is different from group A hormone, and in this case this G protein is called as GQ, and the function actually similar with group A hormone. That means when hormone binds with this receptor, this will activate heterotrimeric G protein, so alpha and beta gamma subunit will be separated, so this active alpha subunit will combine with GTP. Now this alpha GTP activate another protein which is different from group A hormone. In group A hormone, this protein was adenylyl cyclase, but in these cases, this protein which is activated by alpha GTP, and this is phospholipase C. Then this phospholipase C cleave a plasma membrane protein which is PIP2 or phosphatidyl inositol 4,5 bisphosphate, and after splitting, this PIP2 converted into IP3 or inositol 1,4,5 triphosphate and diacyl glycerol. Now, this diacyl glycerol activate protein kinase C, but in case of group A hormone, adenylyl cyclase activate protein kinase A via cyclic AMP, but this DAG, which is a type of second messenger, this will activate another protein kinase which is protein kinase C. Protein kinase are those proteins which phosphorylate another type of protein. So kinase means phosphorylation, right? Now this IP3 which is generated after splitting of PIP2 will be bind with smooth endoplasmic reticulum, and you know smooth endoplasmic reticulum is a calcium storage organelle. So in membrane of smooth endoplasmic reticulum, IP3 receptor present, and after binding of this IP3 into IP3 receptor of smooth endoplasmic reticulum, calcium will be released, and this calcium binds with calmodulin. The hormone acts via G alpha Q and activate an enzyme phospholipase C, and PLC catalyzes conversion of phosphatidyl inositol 4,5 bisphosphate or PIP2 into IP3 and DAG. And what will be the function of IP3? IP3 receptor present in smooth endoplasmic reticulum, so IP3 release calcium from smooth endoplasmic reticulum, so intracellular calcium level will be elevated. And this calcium also acts as second messenger. Calcium binds with calmodulin, and calcium calmodulin complex activate enzyme called calmodulin dependent protein kinase which activate many downstream protein. Now another second messenger which is formed by action of group B hormone is diacylglycerol. Then this diacylglycerol activate protein kinase C, and which phosphorylate many protein. So this is mechanism of function of group B hormone. Present 4 families of G protein, and the G protein which activate adenylyl cyclase, and this is known as GS, and this causes conversion of ATP into cyclic AMP, so this will generate cyclic AMP as second messenger. And another important G protein is GQ, and this GQ will be activated by group B hormone, so this GQ activate phospholipase C and which break PIP2 into IP3 and DAG. Another 2 type of G protein is GI, and this GI inhibit adenylyl cyclase, and since adenylyl cyclase elevate cyclic AMP level, but this GI inhibit this adenylyl cyclase, so cyclic AMP level in the cell will be lowered, right? And another type of G protein is G12 protein, and its function is not discovered still. Okay, actual mechanism by which G protein coupled receptor acts via group A and group B hormone. And in next video we will discuss about receptor tyrosine kinase. So thank you, and that's it.